I think it's probably a good time to get started. I'm very delighted to welcome three speakers today at today's lunchtime meeting, giving us a talk called The Zombie Effect, a re-analysis of 3 NMAs of antidepressants for the treatment of depression in children and adolescents. And our three speakers are Martin Plöderl, who's a clinician, a clinical psychologist working in Salzburg, and he's also a member of the expert panel of Austria's national and local suicide prevention program. Florian Naudet is a psychiatrist who's currently professor of therapeutics at Rennes University and a senior member of the French Institute in France. And thirdly, Richard Lyus, who's an ST6 child and adolescent psychiatrist about to complete training here in Cambridge, who has interest in the evidence base for psychiatric medication and also reducing the use of restrictive practices in the child and adolescent inpatient units. So I'm very happy to hand over for around a 45-minute presentation, and then we'll have time for questions afterwards. You're very welcome to deliver questions to the chat as they occur to you on the way through, and then we'll come to all of those questions at the end of the talk. Questions are very much encouraged, but we'll save them all for the end. So over to the team of three.
Thank you very much, Hilary, and thank you very much, Ollie, for this opportunity and for arranging. Let me just get my slides up on my screen. Okay, so, first of all, I should say that obviously today you have myself, Florian and Martin, but our fellow researcher, Gert van Valkenhof, couldn't join us today. He works at the Cochrane Institute, and his input was essential in producing the data and analysis that we're going to talk about today. None of us have any relevant conflicts of interest to declare, we think, related to our presentation today. And an overview of our talk today is going to be that because we're focusing on reviewing the 3 major network meta-analyses that have looked at the use of antidepressants for the treatment of depression in children and adolescents. And so my task initially is to set out the evidence in those 3 NMAs and how they have informed clinical guidelines, and then also to illustrate that there's an important and clinically meaningful discrepancy in the findings across those NMAs, specifically for the efficacy of fluoxetine as assessed using clinician-rated depression symptoms. A bit of a spoiler alert, part of our analysis revealed that one of the studies in 2 of the NMAs was a highly problematic outlying study with impossible results that could be classified as a so-called zombie trial. And if you haven't heard of those before, the good news is that Florian is an expert on research integrity and evidence syntheses and zombies, and he will, after Martin has talked you through the process of our investigation of the discrepancy and identifying the zombie, he's going to talk to you about the impact of those zombie studies in the medical literature more generally. And then I'll very briefly say something at the end about what I think the clinical implications are for child and adolescent psychiatry of our research. Just to be very clear at the outset, we're focusing today on the use of medication for depression. We're focusing on children and young people, and we're almost entirely talking about fluoxetine, although we'll comment on other interventions very briefly. And we're also focusing on, in terms of efficacy outcomes, on clinician-rated depression symptoms and not on other outcomes that can be used to assess efficacy, although it's worth noting that our findings here are very much in keeping with the other efficacy endpoint findings. So why are we focusing on fluoxetine? Well, that's because, as you may know, fluoxetine is almost universally in guidelines in Europe and North America recommended as the either first-line medication when medication is to be used for treating depression in children and adolescents. And in some cases, it's the only medication that is specifically mentioned. And the reason for that, sorry, those slides that I meant to delete those, let me skip forward here. So, and the evidence on which those guidelines are based comes largely, not exclusively, but largely from these 3 major network meta-analyses that have been published in the last 10 years. And if you look at the guidelines I've just shown you the Maudsley guidelines or the American Academy guidelines in the USA. They all point to these 3 NMAs to support their recommendation for the use of fluoxetine as the first-line medication treatment. So what did these 3 NMAs show? The first was published in 2016 by Cipriani and colleagues, and they included 34 studies. They looked at all antidepressants, so not just SSRIs and more recently developed drugs, but also older medications like tricyclic antidepressants. And they found that all drugs were statistically equivalent to placebo, except for fluoxetine. And for fluoxetine, they reported an effect size as an SMD of 0.51. Importantly, although that SMD is often quoted in the literature and is mentioned in some of those guidelines when they're talking about why they recommend fluoxetine as first line, they often omit to mention some really important contextual information for interpreting that effect size, which is firstly that it was very imprecise. And you can see here that the uncertainty interval ranged from almost 1 to almost 0. And because of that, and because of other limitations in the data, the authors gave that finding a very low confidence rating. So what does that mean clinically? It means that we have a moderate effect size, but it's very uncertain, and fluoxetine, based on this information, could be better than placebo, or it could be equivalent. A subsequent network meta-analysis by Zhao and colleagues, published in Lancet Psychiatry 4 years later in 2020, looked not just at medications, but psychotherapy too. So they had a bigger network of interventions for comparison. But they included almost exactly the same medication studies. So there were 36 studies just of medication. And they found very similarly that all drugs and all psychotherapies, in fact, were statistically equivalent to placebo, except for fluoxetine. And they found a almost identical SMD of 0.5, but again, imprecise with a wide uncertainty interval. And because of that and other factors in the data, they gave it a very low confidence rating. So based on those 2 NMAs, you could make an argument in terms of your clinical interpretation that you could justify the cautious and speculative use of fluoxetine as a treatment in this context, and it might be better than placebo, or it might not be. But then in 2021, there was a NMA published under the auspices of the Cochrane Group with Sarah Hetrick as the lead author. And they focused only on what they called new generation antidepressants. So essentially, they excluded tricyclics and any other older medications. And what they reported was in some ways quite similar, but in some ways different in clinically meaningful ways. 2 important differences about the conduct of this NMA was that, firstly, they didn't report SMDs. So it makes it initially difficult to compare the findings across the 3 NMAs. They reported the raw difference in the depression rating scale between drug and placebo. And they focused on the most widely used rating scale, which is the CDRSR, the Children's Depression Rating Scale Revised, which ranges from 17 to 113. So it's a range of 96 points. The second thing they did differently from the beginning was that they predefined a threshold of clinical equivalence. And they reviewed the literature on depression rating scales, and they decided that plus minus 5 points on the CDRSR, which was roughly equivalent, depending, of course, on what standard deviation you use. But using the typical standard deviations from these studies, that would equate to an SMD of about 0.35. And they very helpfully set out in advance that if something fell within that range of equivalence, they would make certain statements in their conclusions about the efficacy of the medication, namely that it would be clinically equivalent to placebo. And what they found was that fluoxetine was 2.8 points better than placebo at improving depression symptoms. And Martin identified that the authors, although they didn't report an SMD, they did report a standard deviation for their pooled data set, which was 14.4. So if you use that standard deviation, you can calculate an effect size for fluoxetine from this NMA of minus 0.2. So what you'll probably notice straight away is that that's considerably smaller than the SMD that was found in the previous two studies. And you also might notice, if you look at the uncertainty intervals, that it's a much more precise finding with much tighter uncertainty interval ranging from minus 0.28 to minus 0.1. The authors also gave this finding a higher confidence rating. They rated it as moderate compared to the very low rating for fluoxetine's efficacy on the previous two studies. So the key points about the three NMAs are that interpreting this from a clinician's point of view, you look at Cipriani and Zhao and you think, well, all drugs are statistically equivalent to placebo except fluoxetine. So in terms of what I feel able to prescribe based on these studies, it's only fluoxetine, and I'm using that on the basis that it could be equivalent or could be better than placebo. But the Hetrick study points us in a different direction clinically, because all drugs, including fluoxetine, fell within the author's predetermined range of clinical equivalence. And it's worth saying that sertraline and duloxetine had, similar to fluoxetine in the Hetrick study, they did statistically separate from placebo, but only just. So the only medication or intervention at all for which there seems to be some evidence to support cautious and speculative use would be fluoxetine, if you were to be guided by the Cipriani and Zhao NMAs. And if you look at those on this forest plot here, and we put all of the results together, what you'll notice looking at the dark black lines, which is the overall results for the three NMAs, you've got SMD of minus 0.5 for Cipriani and Zhao, and minus 0.2 for Hetrick. And what you'll also notice, if you're familiar with NMA methodology, and it was this discrepancy between the three NMAs that first drew our attention to look more closely at these studies. But it was also the fact that if you look in the supplementary material for the Cipriani and Zhao NMAs, you can find the direct head-to-head comparisons for all of the fluoxetine versus placebo studies. So without the effect of indirect data, and Martin will talk briefly about NMA methodology in a second. But what you'll notice is that not only is there an important difference between the overall results between Cipriani and Zhao versus Hetrick, but even within the NMAs themselves, the direct result is markedly different in Cipriani and Zhao from the indirect result. And the direct result in both those NMAs is actually in much closer accord with the findings of Hetrick. So that pointed us to the fact that there was something problematic going on in both of those studies, potentially with the indirect data. I'll hand over to Martin now, who's going to talk to you in a bit more detail about how we investigated this discrepancy.
Yeah, good afternoon and hello from my side. So the first question that we asked ourselves, was there just a different selection of studies in these three network meta-analyses? We already heard that at Cipriani, they used all antidepressants, the older and the newer generation ones. And Zhao, they used all antidepressants, but they also investigated psychotherapy trials and the combination of psychotherapy with drugs. And in Hetrick, they only used new generation antidepressants. And since Hetrick is the most recent trial, the first question that arose was, can the inclusion of more recent trials explain the discrepant findings between the more recent Hetrick and the older Lancet meta-analysis? So the first thing that we did was to do our own meta-analysis of all the available studies where fluoxetine was compared to placebo. And on this graph, you see on the x-axis the year of publication of the individual studies. On the y-axis, you see the effect size. 0 would mean there's no drug placebo difference, and anything below 0 is in favor of fluoxetine. And as Richard already mentioned, here you see the dashed horizontal black line. This is the threshold of clinical importance, which means that anything between 0 and this area would be a clinical equivalent with placebo. You can see that the initial trial, so each trial is a dot, and the bar is the area of uncertainty, the imprecision of the estimate. So you can see that in the early trials, So, and here you can see that in Hedrick, there were only 2 novel trials that were not included in CHOW. You can also see that these early trials did find some clinically meaningful effects, and later on, the effect was much lower. What you can see here is in the gray shaded area is the meta-analytic aggregation of the evidence. So for each new trial, we did a new meta-analysis, and here you can see that the efficacy is kind of moving into the area of clinical equivalence with placebo. Okay, so this is what we found, and in our publication, you can look up for a different explanation why efficacy can diminish or reduce so much. This is a phenomenon that's not only in antidepressant research, but also found in other fields. So we found evidence for this novelty bias. However, this inclusion of more recent trials cannot explain the discrepant findings between the Cochrane and the Lancet meta-analysis, because already in 2015, you can see here, this was already in Cipriani actually, that efficacy kind of converged to the final level. Afterwards, the inclusion of more recent trials only led to a more precise estimation, but not in a substantial difference in the point estimates of efficacy. So something else must be going on, some error or something else needs to be found to explain this difference. So we looked for errors, and in fact, we found the so-called zombie. To explain what we found, I have to do a little, give you a little background in network meta-analysis, what happens there. So in classical meta-analysis, the figure that you just saw, you only use direct comparison. So you, for example, you compare a trial on fluoxetine versus placebo. And in this trial, for example, you find an effect of 0.3 standardized mean difference. In network meta-analysis, you go one step further. For example, you can include a trial that compares fluoxetine to another drug. And for example, you see that fluoxetine is superior to this other drug with 0.2. And then you have a trial where this other drug was compared to placebo with an effect size of 0.2. And this you can use to create indirect evidence, which would be in this case a difference of 0.4. So you just add up these differences. And what is important, crucial in the interpretation of network meta-analysis is that there shouldn't be discrepancies between direct and indirect evidence. So once you see these differences, you should be careful. So very fundamental assumptions of this analytic approach are not met. And indeed, as Richard already showed you in his figure, there is a substantial difference between the direct comparisons of fluoxetine with placebo, which is 0.3, so the typical effect size you find in antidepressant literature, and the indirect evidence 1.4. You usually do not find such large effects in psychiatry at all. And actually, this led Cipriani and Chow downgrade their evidence to a low certainty rating. They also said that this is somehow related to the fluoxetine nortriptyline placebo comparisons, but the difference or the discrepancies remained largely unexplained. So we looked at the results table, all those different direct and indirect comparisons of all drugs, and we found effect sizes of around 4. So, and this is just not possible in evidence-based medicine. This is too large for any field. And you only find such extreme effect sizes in very extreme comparisons, such when you ask people if they prefer chocolate over feces. And to give you a graphical depiction, these are all the effect size of the different comparisons. And here you see one outlier. It's Atari 2006. So we took a closer look at this trial. It was an older trial, a small trial, only about 40 participants, and it was a trial where fluoxetine was compared to nortriptyline with an effect size of 4.2. So this is not possible. And we found out this single trial caused the large indirect effect for fluoxetine. You see this here in this figure. We also found some mismatches and errors in the data of this trial. So for example, when we calculated effect sizes based on the endpoint scores, depression values at the end of the trial, we came to a much lower effect size estimation. And there were other problems in this trial. For example, they replaced dropouts with new cases. The p-value didn't match the t-test results, and there was no ethical approval mentioned. We tried to contact the authors for clarification, but received no response. And we also applied the new Inspect SR tool, where you can judge the trustworthiness of a trial. And overall, we came to a low certainty in the trustworthiness of this trial. And yeah, I think we detected this zombie. So a logical next step was then to see what would the analysis be without this zombie trial after we disinfected the analysis, so to say. To do so, we requested data and code from the authors, and this was admittedly a sobering experience. We did not receive responses or only after multiple attempts. The data was not available from the authors, and we only partially received data from Hattrick, who said they couldn't access their data anymore, which is quite striking for a Cochrane publication from 2021. We fortunately found then the data for Cipriani and Chow online in another publication, secondary publications on these trials. And the code was not available. And also, we had to do the statistics and statistical coding ourselves. And this is where Gerd was super helpful because he's an expert in Bayesian network meta-analysis. So this is what we found. The three different network meta-analyses are in different colors. On the top row, you see the original results of the meta-analysis. The second row is the reanalysis without the zombie, and the second row is our reanalysis with the zombie, and the third row is the reanalysis without the zombie. And there were two main findings. First, our reproduction worked quite well. We couldn't perfectly reproduce the original findings, but we were close. And conclusion is that we should have had access to the code and the data to be able to perfectly reproduce the findings. And the second and most important finding was that after the exclusion of the zombie, all the three network meta-analysis results were much in alignment. There were no real disagreement anymore. And there was also the statistically significant and major discrepancy between direct and indirect evidence with the zombie disappeared afterwards and was not statistically significant anymore. So to give you an intermediate summary before Florian will talk more about zombies. So the guidelines we are using in clinical practice heavily rely on the three network meta-analyses by Cipriani, Chow, and Hattrick. But those network meta-analyses have quite different clinical implications for fluoxetine. So in the first Lancet meta-analysis, they conclude that this drug is maybe clinically beneficial with a low certainty evidence rating. Whereas in the later Hattrick-Cochrane meta-analysis, they concluded that the efficacy of fluoxetine is equivalent to placebo with a moderate certainty evidence rating. We could show that the earlier Lancet network meta-analyses are infected by a zombie, and that after the exclusion of the zombie trial, fluoxetine is now clinically equivalent with placebo even in the earlier meta-analysis. The results of all analyses agree. And strikingly, we could find that even a small zombie trial can have a large impact in the overall rating of a really large meta-analysis. Also, surprisingly, the zombie was not detected even by experts. So the authors of the two Lancet meta-analyses are really well respected and esteemed and experts in the field. So we think that our reanalysis is another example that post-publication peer review is important, and to do so, it requires the sharing of code and data, and actually there's no reason to not share data and code for meta-analysis. So then I would like to hand over to Florian.
So basically, the next part of the story is that we tried to submit this to the Lancet first, and we contacted their, editors, and especially they have a good editor in charge of integrity. And I will start with a quote from Richard Horton, who is the editor-in-chief of the Lancet, who said that the case against science is straightforward. Much of the scientific literature, perhaps half, maybe simply be untrue. And I think he is an expert for saying that. And actually, this was the outcome. So they sent our study to peer review, and it was peer reviewed by colleagues, and the feedback was interesting. And I tried to summarize here the feedback. The first feedback we got was that, okay, Atari, the study by Atari was problematic, but it met all inclusion criteria for the meta-analysis. And you know that in a meta-analysis, you should be exhaustive. We got the feedback that there was little evidence that Atari was made up. So people agreed that it was problematic, but little evidence that it was made up, totally made up. We got the feedback that the inconsistency, as told by Martin, was explicitly acknowledged in the systematic review and meta-analysis by Cipriani and Zhu. They said, we have inconsistency. They don't really explain why, but they said that it was here. So basically, the meta-analysis is not wrong because it's acknowledged. And the discussion emphasized substantial uncertainty in effect estimates. And this was reflected in the, rating of confidence in the evidence. And then we got the feedback that clarification of the problem with Atari should not be done with the Lancet, but it should be done with the journal that published the study at the beginning. So that basically what we received as a feedback. And the conclusion was that the editor did not see a need to change either of the papers published in the Lancet and the Lancet Psychiatry. First point, okay, the study was problematic, but the meta-analysis was exhaustive, and this is a principle in meta-analysis. You should aim for exhaustivity. Little evidence that Atari was made up, of course, but we lack access to IPD of Atari. And when you are looking for zombie trials, you are always looking at the smoking gun, and IPD are the smoking gun. So, but they don't share the data, so it's difficult to say. Of course, we can only rely on what is written in the very inconsistent paper. Inconsistency was acknowledged, but not the reasons. And then there is this question that whether we should consider that low evidence for a modest effect is the same that moderate evidence for a small effect. So meaning that have we already reached a different conclusion, and this is open for discussion, and maybe we will have different interpretation on that. And of course, we try to contact the journal publishing Atari, and nothing happened. So I think Richard told you about a little bit evidence-based medicine and the importance of systematic review and meta-analysis. Those are often seen as at the top of the evidence pyramid, but meta-analysis are synthesis approach. They are not like primary research, they are secondary research. And they are looking at the study. So they are looking at all the studies. They should be exhaustive. And in the pyramid, we have, of course, the perfect studies, and we have also the other studies. And for meta-analysis, we are used to that. We are used to use, risk of bias assessment to understand if a study is at risk of bias or not at risk at all. So we have some tools for that. We have also then tools to grade, quality of evidence and strength of recommendation based on meta-analysis, based on methodological aspects of the study, based on several parameters of the meta-analysis, whether we have inconsistency, how much we are, estimating the effect with precision. But this is for, studies that are trustworthy. And for meta-analysis, we don't have a true tool to detect studies that are not trustworthy. This tool is being developed. Martin discussed the Inspect-SR tool, and hopefully it will be published soon. But still, we don't really have a tool to judge whether a study can be trusted or not. And actually, there are, in the literature, studies that are not so trustworthy. We have, first of all, a lot of studies published in the literature that were, subject to good clinical practices inspection by health authorities. And we tried to make a survey of all these studies in which health authorities, here it was the European Medicine Agency, found problems within the trial when they performed their good clinical practice investigation. And actually, it's true that they found sometimes some problems, and those problems are almost never reflected in the published literature. So if you just focus on the trial published in the literature and what is said in those reports, you will miss sometimes very serious issues. And in the field of antidepressant research for adolescents, you have this very famous study, study 329, that explores the efficacy and harms of paroxetine and imipramine in treatment of major depression in adolescents. And actually, this trial was very poorly reported, and it became a case study because it was one of the first very big study to be reanalyzed based on the IPD data. So you got the IPD. But all those studies, the one I told just before, this one, are studies in which you have some reporting issues. Sometimes you have some centers that are problematic, but not the full trial. But we have also those trials that we call the zombie trials when we are very, very beneath the evidence floor. I will give you a few examples. Here is a trial about Artemisia annua and Artemisia afra, tea infusion. So Artemisia is a plant. This was a trial published in Phytomedicine. And they try to see whether tea infusion of these drugs could help on schistosomiasis. So it was a large clinical trial. You can see 800 participants. So I won't go in all the detail about this trial, but I will show you this table that was from the supplement. It was in the supplementary material. And please, just have a look at this table. It reports on the adverse events among the participants in the trial. And if you have a close look at it, I think you will see something. Actually, I always showcase this to my colleagues when they ask me what I do to teach zombie trials to my students. I say I show that. So a few years ago, I showed this to my dean and I say, please find it. And actually, my dean, who is an expert in clinical pharmacology, didn't spot anything. And maybe you have not spotted anything in this trial. But then I come with the next slide, this one, because I put some red. And if you look at this slide, you will spot it directly because all adverse events or almost all adverse events are multiple of 5. And it's not possible that in a trial, all patients come with an adverse event by group of 5. They don't come with a cluster of participants with the same problem at the same time and so on. So this was the kind of, things that are totally impossible we can find in those zombie trials. Here is the second trial by the same group because often you have, it's like in zombie movie, you have the first zombie movie and then you have the sequel. And the sequel is almost the same, sometimes a little bit less fun, but still it makes money. So for zombie trial, it's exactly the same. So you have here a new one about Artemisia annua and Artemisia afra. But this time it was in malaria, so another disease. Okay, well, it came with the same number of ethical approval. This is the first problem. But then you have the table with the adverse events, and hopefully now you have this in your eyes and you can spot easily the problem with the adverse events. Adverse events never occur only or almost only by multiple of 5. Here it was exactly the same data pattern. If I had more time, I will give you much more detail because the study is much more funny than that. Here, another study. It was found by Dorothy Bishop. Maybe you know Dorothy Bishop. It's a colleague from UK. She was interested in looking at this trial, and she asked me to have a look. And at the beginning, I hadn't have an access to the paper because it was under a paywall. And in France, sometimes we don't have access to all paywalls, unfortunately. So actually, I looked at clinicaltrial.gov and I found that. So it's a trial that studies the effect of low-level laser therapy in autism spectrum disorder. And it was the primary outcome. It was a mean change from baseline to study endpoint. We had 21 patients in the laser group and 18 patients in the placebo laser, which is something quite strange, a placebo laser, but it can happen. But we have 20 and 18 patients, 19 patients. And here in this group, we have, a pre-post difference of minus 14, which is something that is possible. Where here the standard deviation. But if you look at the, placebo laser, you have a difference of 0.037. This is not really expected when you assess a pre-post change, because often with these scales, you can expect some regression to the mean and specific factors and so on. That will 0.37 is not that much. And especially if you look at the standard deviation, you can see that it's small also in comparison with this one. So very low variability in this group. So you know that when you will have access to the paper, you will have to look at this kind of stuff. And then we had access to the paper and we found inconsistency. So when you are looking at zombies, basically you are looking at whether there are inconsistencies in the paper. So for instance, we have the global score on ABC scale here and the subscale scores. And global score was 30.5 plus minus 6.7. And here they give the same scale, but the subscale score, which is just a part of the scale. And on the subscale score, you have exactly the same as the global score. So it's an inconsistency. Of course, an inconsistency does not make the paper a zombie because, you need to find much more than one. It can occur by mistakes. We are all making always mistakes and errors in our papers at a rate that, for me at least, I cannot figure how much I can do this, but it's a big problem. But then you look if there are systematic problems. And if you remember, I told you we need to look at what happens in the placebo group. So here is what happened in the placebo group on the different subscale score of this ABC global scale. And global score, you see there is a very little difference, which is very coherent with what we've seen in clinicaltrial.gov. But for the subscore, here you have no variability before and after. Here, no variability. This is what we can call an excess of homogeneity. And when you have this kind of excess of homogeneity, because there is still always, inter-individual variability and also intra-individual variability. Tomorrow, I will not be the same as I was today. At least if you look at my parameters like blood pressure and stuff like that. This is medicine. And what is good, I told you we need the smoking gun. And what is good is that the authors were very transparent. And when Dorothy Bishop asked for the data, they share the data. So here is the data, in the test group. And basically, the data are what they are. One striking point is that there is no missing data, whereas you are always missing data in clinical trials, but still, maybe they have a very good follow-up and so on. But here is the data in the placebo group. And this is again the same. You can see that for several participants, the line is a patient. The data is exactly the same at baseline, week 2, week 4, and week 8. On scales, scales, there is also, even if the patient is the same, scale, you have, measurement error by the evaluator. Even if I do the scale just before eating and just now after eating, I'm not sure I will rate things exactly the same, especially regarding exams that looks talk about food. So this is a smoking gun, and this is what we didn't have with Atari and Al. Here in another example, you will see a fun trial because I was speaking about that with a Cochrane dermatologist involved in Cochrane, and they give me an example. So we had a look at this example, and we found that here it's a comparative randomized clinical trial evaluating the efficacy and the safety of tacrolimus versus hydrocortisone as topical treatment of atopic dermatitis in children. And here you can look at the graphs, you can look at the error bars, and if you look closely, you will see that there are errors in the error bars. You know, those error bars were written by hand, you know, with PowerPoint. Those are not done by Excel, they are done by PowerPoint. You know, it's not perfectly here. Here it's not totally, totally straight. So that's exactly what we can try to find when we are looking for zombies, this kind of stuff. And this is what InspectSR tried to help to do. Hopefully, if people who are committing this kind of zombie trials are not getting more and more adapted and will change their way to produce this. And what we gave you as information about these fluoxetine trials needs to be a little bit looked with an bird eye view. And people have done some kind of systematic investigation of meta-analysis, including not only zombie trials that are, haunting the literature, but, zombie trials that were retracted. Sometimes retracted trials are cited and included in meta-analysis. And they tried to look at how much it changed the effect size. And you can see that in gray, it was more than 30% change on the effect size. In yellow, it was between 20 and 40% change in the effect estimates. So if we look only at gray and yellow, it means that a substantial part of the meta-analysis included, each square is a given meta-analysis that included a zombie trial. So a substantial part actually had a change in effect size that were so above 20% in the effect estimates. So for us, we were at least, I think, 50% of the effect estimate. So it was one big outlier. But actually, this is a problem, infestation of zombie trials. And all those meta-analyses are still in the literature. And a new study, an important initiative, tried to identify the contamination between, trials that are problematic, zombies, evidence synthesis, and then guidelines. And you know what? Trials that are problematic are making their way in evidence synthesis and guidelines. So meta-analyses are exhaustive, of course, but sometimes being exhaustive can be problematic, reproducible, quantitative, of studies addressing the same question, but they are not always fit for purpose to deal with zombie studies within our current research ecosystem. So now I will give the floor to maybe Richard for concluding, and I think we are almost done.
Thanks, Florian. I'll just say very quickly in 1 minute, if that's okay, Hilary, just going back to our specific study and the zombie contamination of meta-analytic data sets that we've described. In terms of clinical practice for child and adolescent psychiatrists, I think the findings really need to be taken into consideration. If you look at the forest plot of our Re-Analyses here, and look at the reproduction of the Cipriani and Zhao NMAs with the exclusion of the zombie trial, Atari et al., you can see that the overall estimate for the efficacy of fluoxetine is much smaller and in accord with Hetrick, and that the point estimate and most of the uncertainty interval for both the NMAs falls within the zone of clinical equivalence. Next slide, please, Florian. So I think the clinical implications are the evidence for fluoxetine was already not terribly robust for this indication. It was a moderate effect size that was very imprecise and of low quality. And our analysis shows that the moderate effect that was found in the earlier Lancet NMAs was driven largely by invalid data from a single zombie study, and that excluding that from those data sets gives you an effect size that's comparable to that of the Hetrick Cochrane NMA, which is an SMD of about 0.2 to 0.3, which is roughly equivalent to about 2 or 3 points difference on the 96-point Children's Depression Rating Scale. So an extremely small difference. I think this falls well within any reasonable quantitative definition of clinical equivalence. And given that this finding for symptom reduction fits in with other efficacy outcomes, such as effect on function, self-rated depression symptoms, suicide-related outcomes, I think it's very hard to make an argument that there's evidence to support its use as a first-line agent for this indication, or indeed at all. And I'll finish there.